I'd like to introduce Denise. Titian, HighRes and Genedata have been working with Sygnature to help them get their HTS system underway, and Denise is going to tell the story of the processes that they do and how the project went. So Denise, please.
So I'm going to talk to you about the launch of HTS at Sygnature Discovery. This was supported by Titian Mosaic, HighRes Biosolutions and Genedata. So just to outline what I'm going to talk to you about this morning. So why HTS? Why did we want to set up this capability? Very much to do with hit identification and to complement Sygnature Discovery's current suite of hit finding approaches. Then I'll talk to you about building the capability from scratch. It's not just a robot, there's so much more involved in that. I'll talk to you about how we designed our library, the automation obviously, compound tracking and storage, and data analysis and fidelity. Then I'll talk to you briefly about a case study that we got some data for, HPK1, and it was our pilot HTS, and then I'll summarise. So before I launch into that, I just want to tell you a little bit about the Sygnature Discovery group. So Sygnature Discovery was founded in 2004 at BioCity in Nottingham in the UK. We're a provider of high quality, integrated or single discipline drug discovery. We've got a strong track record in drug discovery with 16 compounds delivered into the clinic to date in phases 1, 2 and 3 since 2011. We have more than 250 staff in the disciplines of medicinal chemistry, bioscience, DMPK and computational chemistry. In 2018, we expanded our capabilities and acquired Renasci. Renasci were again founded in Nottingham at BioCity in 2001, and they're a company that specialise in conducting in vivo studies with more than 30 scientists. They've facilitated more than 30 compounds into clinical development and 10 drugs onto the market. So as you can see, we're predominantly based our HQ at Nottingham at BioCity, but we've also expanded out to Alderley Park as well in Cheshire. Okay, so what you came here to listen. So why HTS? To this audience, I think it's obvious, but HTS is a well-established process in the drug discovery industry, used to rapidly aid identification of novel chemical starting points. It's a combination of medicinal chemistry, biology, informatics and advanced automation technology. Because of the complexity of the process, many factors in addition to the biology influence the quality and outcome of a HTS process. One of the most key components of this is access to a high quality lead-like compound library. So with all of these things in mind, at Sygnature we've set out to design and build a HTS capability that addresses all of these points above. As a CRO, the capability we've designed had to be up and running as quickly as possible. So with that in mind, we've worked with industry leaders who've provided solutions that will ensure that we can rapidly and more importantly be sure of the quality of the hits and the data that we provide to our clients. So hit identification. The focus of hit identification is to apply the most appropriate hit finding approaches to successfully identify hit series with the best chance of developing into drug-like compounds. Before we added HTS, we already offered virtual screening. We've done many medium throughput screens. We offer fragment screening and we have our own proprietary fragment library. We also use knowledge-based design. And adding HTS to this very much complements these processes. And also there was client demand for this service as well, meaning that we could offer an integrated hit finding approach. As you can see on the right hand side, HTS is still a big contributor to compounds getting to the clinic. So I'll move on to our LeadFinder HTS library. So this library is a broadly applicable HTS screening set. It's a 150,000 compound diversity deck. It's got attractive low molecular weight starting points with developable lead-like properties, and it's the all lead-like properties that are important. It gives a good start point in terms of chemical matter, and every molecule in the library has been looked over and curated by one of our senior medicinal chemists. It's a nice library. So the maximum similarity between the compounds has been controlled to maximise diversity. We have good chemical purity, and we monitor the QC, the stability, with ongoing QC. The library has been designed to allow rapid follow-up, either in-house chemistry or we can purchase in solid. The library was, again, one of the criteria for designing the library was that there were significant amounts of solid available for purchase should we need to. So in addition to this starting point that we've built purchasing the library, we're also doing an ongoing internal chemistry approach to build on this library. So we've got internal proprietary compounds that will be added to the library with time as we go through. So very much with this library is a focus on a rapid follow-up of any hits that come from HTS. So to the automation, this is kind of where I'm probably more comfortable. So HighRes Biosolutions designed and built the automation using their A-cell benchtop robotic arm for us. The system offers high flexibility to scale and reconfigure the peripheral equipment as the bioassay or the technology dictates. So the automation's controlled by Solario. This is dynamic scheduling software that allows flexibility to adapt the kind of screen that we run on the system. So the whole automation system has been built around lab-site echoacoustic technology, and this means that we're going to have high precision in our dispensing. It also means that we don't go through our compound library too quickly. Nanoliter quantities of compound are used. So the system's modular, it's expandable, and it's capable of running both cell and biochemical assays. And you can see some of the pictures of the system at the bottom there. Okay, so moving on to compound management, and this is probably where I'm less comfortable, but again, it's all part of the capability and the solution that we've built. So fully integrated informatics platform is needed to ensure high data fidelity and compound tracking. So in our processes, assay plates are ordered using Titian Mosaic sample bank compound management software, and this is linked to our HighRes automated system. The LeadFinder library is housed in a custom-built compound store. This keeps them at low relative humidity at ambient temperature and makes sure that we keep our compounds in good condition and maintain the integrity of the compounds. So that the actual store, you may or may not be able to see from the image on the top right-hand side, it houses HighRes stackers. So we simply go into the store, take out the stacker, and then put it onto the HighRes system, onto our Ambi store. So it basically minimises human intervention, people shuffling plates, getting things in the wrong order. So compound integrity and depletion of the library plates actually are monitored in real time using feedback from the echo on the HighRes system. This is all passed into Mosaic. And in addition to this, we do regular QC of the library. So to date, we've measured part of the library, and it's an ongoing process, but 90% of the compounds within the library, more than 90% pure by LCMS, and this is from a 15% sample of the library. And with time, we'll do all of the library, and it's an ongoing process, so we'll keep doing this with time so we know that the compounds are in good condition. So to data analysis, again, important part of this story. Information technology systems have to be able to handle and interrogate large, complex data sets. So on our system, the plate files are uploaded to Genedata Screener. The plate barcode automatically pulls in the sample ID and compound location from Mosaic, again, ensuring data fidelity and no human intervention. The upload of screening plates to Genedata is really quick, it's rapid, and it allows rigorous interrogation of data by plate, by batch, by the whole screen. We found Genedata to be intuitive and straightforward to use. It's been really straightforward. Okay, so now I wanted to talk to you a little bit about how we went about putting this, this capability together. So timelines have been pretty tight for the project. We had to get it up and running as quickly as we could. So in the first 6 months was basically the design process, the design phase. So this was an iterative phase, particularly with the automation where we needed to reach a solution from a design and also a cost perspective as well. So at the end of the 6 months, the orders were placed for the automation and all of the software. So in the next 6 months was very much a process for us at Sygnature to make sure that we got our infrastructure in place and ready. So this covered things like getting all the services in place, ready for the installation. Obviously, this is an IT heavy setup, so we needed to make sure all the servers and the databases were all in place, ready. There was lots of iterative conversations with the vendors to make sure that we'd got the right things in place, ready for installation. And also, obviously, we had to do a little bit of lab remodeling, knocking out some benches to make space for the automation. Okay, so at the end of that 6 months, the HighRes system turned up at our labs, so I'm going to call that month 1. So basically, it turned up, we unpacked it, I say we, the HighRes guys, they installed it and recommissioned the robot. That went well, so into month 2, once we got the robot up and running, then we looked at the Mosaic and Solario order broker COB integration. So this was the interface between Titian and HighRes. So we spent a week with HighRes on site and Mosaic on site, actually physically running orders through the system. So we ordered them through Mosaic, sent them through to the automation and physically tested them on the system to make sure it all worked. And then at the end of month 2, we signed off the automation. So we did the site acceptance. So once we'd accepted the robot, we were basically left on our own to I'll say play with it, but it really was a phase of quite extensive testing on our part. So it was a transition from the test environments into production for all systems. We needed to thoroughly test how the systems were going to handle the protocols we wanted to run. So testing COB protocols, sending them through to Mosaic, troubleshooting and training. Obviously, all of this was dry runs, and we essentially set up scenarios to see if this happened, how would we resolve it. It was, you know, we learned a lot from the testing phase. So month 6, this is when we had our Genedata training, and I think that's a testament to Genedata that, you know, that was probably the last thing training-wise that we did, but we've still managed to get up and running quickly. It's super intuitive to use, and we ran the first HTS on our systems. So now I'll move on to the case study, HBK1. So this was basically, it was a pilot screen, so we could thoroughly stress test all of the component parts of our HTS capability. So HBK1, it's an immuno-oncology target involved in negative regulation of T-cell receptor signaling. The HTS was obviously to identify novel chemical starting points to develop potent selective inhibitors of HBK1. Doing this pilot study was it was useful on lots of levels actually. It allowed us to test out all of our systems. It's given me data so I can come out and talk to people about it, and actually it's a really interesting target as well. So on every level, it was a good thing to do. So we measured the intrinsic ATPase activity of HBK1, and this was used to develop an ADP product fluorescent polarization assay. So you can see at the bottom of the slide the schematic for the FP assay. So this was actually a Bell-Brooks screener assay. So onto some data. So people used to looking at Genedata Screener. These are the graphics you get on the dashboard. So top left-hand side, that shows you the Z primes for the screen. And as you can see, generally it was pretty good, but we did have a few issues from time to time. This is why we did the pilot screen. Top right-hand graph, this is basically the activity or the assay window of the FP assay. And then in the middle, you've got a histogram of distribution of the actives. So on a brand new system, and by system I mean the whole capability, we screened the 150K library in less than 4 weeks. The HTS performance was good, with an average robust Z prime of greater than 0.7. We saw good reliability from all aspects of the system, Titian Mosaic sample bank, HighRes automation, and Genedata Screener. Issues observed were predominantly caused by transitioning from the test areas into production. The multi-drops caused a few plate effects, and we're still working to resolve these. I think the extensive testing we did before starting the screen really helped us to identify processes that might cause issues. For example, we've got barcoding in the COB orders, the compound replication part of it, and actually that can be a little bit, or at least in our hands, we found that a little bit temperamental. So we decided to take that part out, pre-barcode the plates and put them on the system that way. Lots of little things like that really helped to get things as streamlined as possible before we started. So now I'll talk to you about a typical screening data for HBK1. So I'll start with the setup. So that the order is generated in Mosaic, job created in CFM, and then passed through to COB. The robot was then set up and the run validated. So this included source plates, controls, and destination plates. We then move around to the compound management. I've split this up a little bit because we decided from testing that we'd split the compound management and actual biological screening into two parts. So the HighRes system dispenses the library plates. The destination plates are sealed, and the robot typically runs unattended overnight until 7 a.m. So depending on how many plates we were processing, it was typically running between 5 and 9 hours unattended overnight. Someone came in the next day during the working day, and again, this was a decision we made. The biological reagents are expensive, so if there were any problems, we were on hand to rectify them. So during the working day, reagents were made up and the assay scheduled and run on the HighRes system. Again, it was typically running between 4 and 7 hours, depending on how many plates we were processing. So as you can see from those kind of adding it up, typically the systems were running from 16 hours a day, some days depending on what we were doing. So then finally, analysis. So the Mosaic results and Solario logs were checked. The data files were uploaded to Genedata for QC and analysis, and basically we just cycled around that, setting up in the evening before we left for work, left for home, left it running overnight, came in, set the biology up, did the data analysis and that kind of cycle. So finally, in summary, I think that close collaboration with the vendors has helped us to get our HTS capability up and running very quickly, with delivery to initiation of the first HTS in less than 6 months. Some of this was down to the choice of equipment and hardware. So we were very careful with all of our equipment and all of the software to go to industry leaders and industry standard equipment. Basically, we wanted tried and tested solutions. Because we had to get this up and running quickly, we went to the market leaders. I think careful planning and preparation for all stages of the project was required. There's a huge amount of IT infrastructure to prepare that I've probably skimmed over because I'm a biologist, not an IT informatics person. But it was all very important to get this right, in addition to the lab remodeling that we did as well. Where we have had issues getting this this going. We've worked through them systematically with the help of the vendors. All of the systems were tested extensively going live, and I think this was really important. And running the HBK1 test case has been invaluable. It means that we've thoroughly tested all of our systems. So in summary, HTS has been established at Sygnature Discovery in Nottingham in the UK to complement our existing suite of hit finding approaches, offering a more comprehensive integrated hit discovery platform to enhance successful early drug discovery for our clients, and more important, helping our clients to deliver new medicine to patients. This has all been possible with the help of the vendors. And then finally, I'd just like to acknowledge some of the people back at Sygnature, that this has been very much a team effort. So thank you for your attention.